Welcome students to the third lecture of the 4 lectures on glycogen, glycosaminoglycans and glycoconjugates. In this particular lecture, we are going to focus on some of the structural aspects of proteoglycans as well as look into their physiological function, or more precisely, we will look into some of the key physiological functions of proteoglycans. Now, we have already in the second lecture talked about proteoglycans, and we will here focus on the structural specifics of proteoglycans. Now, in case of proteoglycan, we have a core protein, as we can see here in this part of the slide, and to which the glycosaminoglycans are attached. So, this is the core protein and this is the glycosaminoglycan. And here we see the glycosaminoglycan that has been used for this specific example is chondroitin sulfate. This chondroitin sulfate is bound to the core protein through a tetrasaccharide bridge. So these are the individual monosaccharides. So 1, 2, 3, and 4. So there is a tetrasaccharide bridge that is linking chondroitin sulfate, which is a glycosaminoglycan that we looked at in lecture 2, which is bound to a core protein. Now, if we look at the core protein at the junction of this binding, we see that the tetrasaccharide unit is bound to the serine residue. And it has been shown that the amino acid sequence at the binding site of this tetrasaccharide unit consists of serine, glycine, X, and glycine, where X stands for any amino acid. And here, if you look at the tetrasaccharide bridge, the monosaccharide that is linking with serine is xylose. And the xylose residue at the reducing end of the bridge is joined by its anomeric carbon. And the concept of anomeric carbon we talked about when we talked about the linkages in the glycogen structure. So xylose is linked here to the hydroxyl group of serine. So this is one of the specific structural insights of a proteoglycan, where the proteoglycan is bound by a tetrasaccharide unit to the core protein, and the sequence at the site of binding is serine, glycine, X, and glycine, where X can be any amino acid. Now we come to another specific structural aspect of proteoglycan. We have talked about this in lecture 2, that many proteoglycans are secreted into the extracellular matrix, but some remain bound to the cell membrane as integral membrane proteins. And two major families of membrane-bound heparan sulfate proteoglycans have been studied. And these two major families are the Syndicans and the Glipicans. Now, when we look at the structure of Syndicans, we can see that it consists of a single transmembrane domain and an extracellular domain bearing 3 to 5 heparan sulfate and in some cases chondroitin sulfate. So let us look at the structure of Syndicans again. So we have a transmembrane protein. So this is the transmembrane domain, this is the extracellular domain, and this is the intracellular domain. In Syndican, what happens? You have 3 to 5 heparan sulfate, and in some cases you also have a chondroitin sulfate. So you have a transmembrane protein that is linked to 3 to 5 chains of heparan sulfate and sometimes one chain of chondroitin sulfate. And this class of proteoglycan are termed as syndican. What happens in glipicans? Glipicans are attached to the membrane via a lipid anchor, which is a derivative of the membrane lipid phosphatidyl inositol. So this is the typical structure of a glipican shown here in this part of the slide. And you can see here there is a lipid anchor in this part of this particular figure, and this particular molecule is attached to a phosphatidyl inositol. What we need to remember is that both syndicans and glipicans can be shed into the extracellular space after cleavage by a protease or a phospholipase, respectively, and they remain bound to the extracellular matrix even after they have been shed into the extracellular matrix. Now, proteoglycans are involved in cell-to-cell recognition and addition, and in proliferation and differentiation of cells. With regards to cell-to-cell recognition and adhesion, Dr. Nerissa is going to talk more about this when she covers aspects of cell biology in the Foundational Concepts course. Next, we come to another structural highlight of proteoglycan. So we have the glycosaminoglycans chain, which can bind to a variety of extracellular ligands and thereby modulate their interaction with specific cell surface receptors. Now, in case of heparan sulfate, we have two domains that can exist. One is called the sulfated NS domain, and the other one is called the Na domain or the acetylated domain. The sulfated domain consists of 3 to 8 disaccharides. And you can see there, this is the NS domain, where we have 1, 2, 3, 4, 5, 6, 7 disaccharide repeats, and followed by that there is the acetylated Na domain. So generally, heparan sulfate will have an alternate of NS and Na domains. Now, the exact pattern of sulfated and acylated domains varies from one proteoglycan to the other. And also one aspect that you need to remember is that the same core protein can display different heparan sulfate structures when they are synthesized by different types of cells. Now let us look at the function of the NS domain in specific. To elaborate on this, I have used the example of antithrombin. We talked about fleetingly with regards to the function of antithrombin in lecture 2. So this is the heparan sulfate, which we already know has an alternate of NS and Na domain. Now, the NS domain is able to bind to antithrombin. Now, once antithrombin binds to the NS domain, it undergoes a conformational change, and once it undergoes a conformational change, it is able to bind to various clotting factors, and in this slide we have used the example of coagulation factor 10A. So by binding to coagulation factor 10A, antithrombin is able to inhibit the function of factor 10A and therefore prevent the formation of blood clot. So what is the take-home message here? The antithrombin 3 is able to recognize specific sulfated NS domain in the heparan sulfate molecule. And once the antithrombin 3 binds to the NS domain, it undergoes a conformational change, and this conformationally altered antithrombin then is able to bind to factor 10A, which is a serine protease involved in clotting, and by binding to factor 10A, it inhibits its function and therefore prevents the formation of blood clot. Let us now look at how two NS domains are helping in the physiological function of heparan sulfate. And for this also I have used the example of antithrombin 3. So you have the heparan sulfate. This is the alternating NS and NA domains, if you remember from the previous slides. Antithrombin 3 binds to the NS domain, which is the sulfated domain of heparan sulfate. Once it binds to the NS domain, it undergoes a conformational change, which we also looked at when we looked at its binding to factor 10A. Once it undergoes a conformational change, then it is able to bind to thrombin, and this binding brings thrombin close to heparan sulfate. So thrombin has a second domain which is able to interact with another NS domain of heparan sulfate. So what we are seeing here is that the binding of antithrombin and thrombin to two adjacent NS domains of heparan sulfate chain in a proteoglycan brings them into close proximity where they can bind to one another, and thereby antithrombin 3 inhibits the activity of thrombin. Thrombin, for your information, is a procoagulant clotting factor. That means it initiates the formation of clot by binding to antithrombin and to the second domain of heparan sulfate, or more specifically the second NS domain of heparan sulfate. The clotting activity of thrombin is inhibited, and therefore it prevents the formation of a blood clot. Let's take another example away from clotting. So we have certain proteoglycans which can form aggregates, and these aggregates are often enormous supramolecular assemblies where we have a glycosaminoglycan core bound to a single hyaluron molecule. The core protein involved is the protein called aggrecan. So if I look at the structure here, this is the hyaluron molecule which I referred to when I was reading through the slide, and this is the core protein aggrecan, to which we have molecules chondroitin sulfate linked, and this linking happens again via serine residues, and by forming this kind of huge structures which we refer to as supramolecular structures, this particular molecule is able to form or provide resilience or toughness to a particular tissue, and this kind of resilience is observed in case of connective tissue. So aggrecan interacts mostly with collagen in the extracellular matrix of cartilage, and that contributes to the development of the tensile strength and the resilience of that particular tissue, in this case cartilage. I believe Dr. Nerissa will talk about this aspect again when she talks about connective tissue in the part of histology in foundational concepts. Now let us look at another physiological function of proteoglycan. The extracellular matrix is a strong, resilient meshwork. As you can see here, it has a lot of components, and mostly it consists of collagen, elastin, and fibronectin. And some of these proteins are multi-adhesive with several binding sites. And one of these proteins present inside the extracellular matrix is the fibronectin, which has separate domains that can bind heparan sulfate, which is a proteoglycan that you already know by now, and other proteins such as fibrin, collagen, and a family of plasma proteins, which we also talked about briefly, which are known as integrins. So this helps in the formation of the meshwork that you observe in the extracellular matrix. And the key to the formation of this meshwork is the fibronectin protein, which has multiple domains to bind to different proteoglycans and different proteins. Now, how do you degrade proteoglycans? Mostly proteoglycans can be phagocytosized, and then after phagocytosis they are degraded by lysosomal enzymes. And these defects in lysosomal enzymes leads to disorders where proteoglycans cannot be degraded, and this impairment of degradation leads to various clinical conditions, and we will talk about a few in the next lecture in the 4-lecture series. With that, I end this short presentation. I hope you are able to understand some of the structural specialities of proteoglycans as well as understand some of the aspects of physiological function that I mentioned during this lecture. Thank you for listening.